Good morning, students. We're now going to start one most important topic of complete biology, you can tell, that is the protein synthesis. The protein synthesis is a complex process. To understand it, we need to understand it stepwise. So, the protein synthesis is having two most important parts. One is called the transcription. Okay? Other is called the translation. Two things are there. So, what is happening now in that case, the transcription is followed by the translation. Clear? Now, see student, one thing, DNA forming mRNA. This is the transcription. This is what? Transcription. Then mRNA that is formed leading to the formation of protein. This is translation. Understood? So DNA forming the mRNA is transcription and mRNA forming protein is translation. Whole of this thing is called central dogma. That means from the DNA to the formation of protein is central dogma. Now we'll be knowing the transcription first, then we'll be going to translation. First, transcription. Now, transcription, the things that we have done in DNA replication, it remains the same. What happens in the DNA replication? We have found that this is the two strands of DNA. Two strands of DNA need to be unwound first. What we have done in case of replication. So, they will be forming. So, it is like this. It forms what? Here, replicating fork. Replicating fork. And then we are finding, I am coming to the earlier one, finding the RNA primer to come here in this place. This is a leading strand. So here we are finding 3-2, 5-, and here 5-2, 3-. So RNA primer has come. When RNA primer will be coming, it is not a continuous strand in both the part where the old strand is separated. So two strand, old strand are separated. Then what you found? There is the occurrence of the primer just below the old strand, isn't it? We have discussed that in the DNA replication. Then what happens here? DNA polymerase find out the complementary nucleotide. Here there is the occurrence of the nucleotide, and here the complementary nucleotide that will be sitting in the RNA primer will be selected by the DNA polymerase. Got it? Based on the nucleotide that is present in the mother strand. Understood? Now what is happening here? It is a fragment that we named it as Okazaki fragment. There also DNA polymerase will be helping to get the complementary nucleotide. That is okay. But ultimately what happens? It is sealed. Ligase is helping in the continuous strand, making a continuous strand. Here it is continuous, here it was discontinuous earlier. Then it became continuous. Up till here, the things remain the same. Got it? Now what is happening exactly in case of the protein synthesis? The strand newly that is formed, it doesn't coil around the old strand. What we have done earlier, we have found that the strand that is formed it was coiling, is not it? But here what is happening? This strand doesn't coil. That is the difference. And it come out from here. So ultimately, that comes out from the nucleus and goes to the cytoplasm. So we are finding the transcription. What is the meaning of transcription now? Transcription means to transcribe something. Transcribe. Transcribe means what? To give the impression. Just like you are preparing sandesh and other thing in home by your mother. What she does? She takes some clay-made material which may be having the structure of fish, flower or all these things. Over that she put coconut or condensed milk or other materials, is not it? And then she takes it out. When she takes it out, she is finding the structure of fish in that the material which she placed. Got it? So, you are finding two words there. One is the template. What is the meaning of template? Template is the strand which giving the information, message is called the template. The structure, clay structure which is used for making the sandesh is the template. This old strand is the template. Okay. Transcribed, which transcribe and the information that is passed is the sandesh that is produced. Here we are finding this structure. It is the mRNA. Okay. Here we are not calling to be a single strand DNA because it carries the message of the DNA old strand. So here it is mRNA. So that is transcription. Clear to you? So we have completed the transcription of the protein synthesis. In the next video, we'll be going for the translation. Thank you, students.